we're going to start with the easy stuff, right? Functions of the blood. And I don't know where all my bullet points went here, but my guess is they're somewhere off the screen. Is there anything about the functions of blood that need clarification? Is there anything in here that you looked at and you were like, wait, what? That's the blood? No? Okay. Look, she's like, big no. She's like, move it on. All right. So when we talk about the composition of the blood, so common questions I get, do I need to know the percentages? No. You do not need to know the percentages. You need to know the relationships. So which one is highest, which one is lowest, right? I'm not going to ask you for the exact percentage of water in the plasma, but I might ask you which compound makes up the vast majority of the plasma. So you should be able to tell me water in that case. The same with the proteins that are in the plasma, which one is the most prevalent, right? When I'm looking at the other solutes in the plasma, there's no percentages there, but you should be able to identify which things are in the plasma that aren't proteins and aren't water. Does that make sense? Okay, and that's true for the white blood cells and the platelets. You should know that the white blood cells and the platelets make up what they call the Buffy coat, not the vampire slayer, the coat. I know, bad joke. It's okay. You don't have to laugh at all of them. Some of them are worse than others. It's just the way my brain works. So are there any questions about the text on this, or any questions about the figure on this slide? A lot of these first slides are really pretty straightforward things. So everybody's good? Okay, speak now, forever hold your peace. What about this? Are there any questions about sort of what makes up the plasma? Anything that's in here? Again, you should know which pieces are waste products, which ones are nutrients, which ones are gases, those sorts of things, okay? Questions? See, I told you you wouldn't hate this chapter. All right. Who panicked when you saw this figure? You can unpanic a little. I'm going to tell you exactly which ones you need to know, and we'll talk about it, okay? So first of all, we are talking about production of formed elements. What is a formed element? Things that are where? Yes, sort of. So formed elements is a sort of an umbrella term for red blood cells, white blood cells, and platelets. Okay, they're not referred to simply as cells most of the time because, for instance, platelets are fragments of cells. Red blood cells don't have a nucleus, right? There's a lot of variation and difference in them. And so they're grouped in this sort of category of formed element, right? So you need to associate that word with red blood cell, white blood cell, platelet. You also need to know the formal terms associated with them. So red blood cells are erythrocytes, white blood cells are leukocytes, and platelets are thrombocytes, okay? So you need to know those terms. Now, in terms of this figure, what we're looking at here is sort of you could think of this as an evolution or phylogenetic tree of blood cells, right? Except this is not true evolution. The reason I say that is because you can trace each product, which is down here, back to a common ancestor with the other products, okay? So the end products are here. The starting cell that all of those end products come from is up here at the top. And that is your hemocytoblast. You need to know him. Okay, hemo blood, cell, cyto. And what is a blast? So say that again. I heard nothing in the words. I heard sounds. There were sounds. There were no words in those sounds. Yes, blasts build. Right. That's the phrasing that I've always used. Blasts build, sites maintain. Right. So hemocytoblast is a building cell for blood cells. Right. So all of the blood cells, red, white, platelet, in my brain, red, white, purple, all start with this hemocytoblast. Now, the hemocytoblast is special because it is a stem cell of sorts, right? It has the ability to differentiate. We used that term yesterday, right? We did use that term in here yesterday, right? Or did I do that in evolution? This is getting really problematic because I'm using similar terms in two different classes. Did we talk about differentiation? Do we know what differentiation means? Let's start there. Let's go back to that. Somebody tell me what differentiation means. Describe it.
As far as like when a cell, stem cell, like it doesn't have a job yet. So differentiation means that it's assigned a job, a specific job.
Yes, it becomes something specific, right? Differentiation means it goes from being one thing to being something else, right? So stem cells differentiate into different kinds of cells. In this case, our hemocytoblast is going to differentiate into one of two forms. It is either going to become a myeloid stem cell or a lymphoid stem cell. Okay, you need to know those. Now, this is the easy part. The lymphoid stem cell is a very straightforward job. It becomes a lymphoblast, which would be an early, young, right? A baby, right? A baby lymphocyte, okay? And that lymphoblast, all the way down here at the bottom, is going to grow up to be a lymphocyte. Okay, that's easy, right? Okay, now so we're going to now forget about the lymphoid stem cell. He's done. You can take him off the list of things we need to think about. The myeloid stem cell is also a stem cell, and it will differentiate again into one of four major subgroups, one of four blasts, basically. Okay, we're going to start over here with the monoblast because it's easy, right? The myeloid stem cell that differentiates into a monoblast is going to grow up to be a monocyte. Nice and easy, right? Pretty straightforward. Okay, now, what do we have left? We have myoblast, megakaryoblast, and proerythroblast, okay? Let's look at, just to finish them, the myeloblast, okay? So our myeloblast is going to grow up and become a progranulocyte, okay? You need to know that. You do not need to know the myelocyte. We're going to skip those. You do not need to know the band cells. Okay, simplifying things down here. Okay, so your myoblast becomes a progranulocyte, and that progranulocyte will differentiate into basophils, eosinophils, and neutrophils. What do they have in common? Yeah, they're all granulated white blood cells. Okay, progranulocyte, granulated white blood cell. Okay, they are related to the monocyte, which is not a granulated white blood cell. Okay, makes sense? Yep. Okay. All right, moving over here. We're going to skip him because he's my favorite and I like to do him last. Proerythroblast. Guess what it's going to become? A red blood cell. We're going to skip early. We're going to skip intermediate. We're going to skip late. We're going to ignore the extrusion of the nucleus. We're going to think about a reticulocyte, because that will come up later on in other things. And it's going to become a red blood cell. Okay. All of this coming from the myeloid stem cell that came from the megakaryocyte. Sorry, the hemocytoblast. My brain's on. All right, now this guy. This guy's my favorite. I like this guy. He is a megakaryoblast. What does it sound like might be important about that name? Mega is big, okay? These are big cells, and they have to be big cells for a reason. So the megakaryoblast is going to grow up, shockingly, to become a megakaryocyte. Now, this is why I like this guy, right? All of the cells in your body work to help you in some way, right? But none of the other blood cells give of themselves in quite the way that the megakaryocyte does. Because the megakaryocyte literally tears pieces of itself off to make platelets. It literally rips pieces of itself off for your platelets. You don't need to worry about this. Just need to know what happens. Okay. Now, in case you're wondering, I will not be giving you this table and asking you to fill it in. Okay. Everybody's like, oh my God. Yeah, no. This is to help you understand sort of how these cells are all related to one another and sort of where they come from, right? So you should understand sort of their lineage, and you should understand their relationships to one another, but don't feel like you have to be able to recreate this table. That's not going to happen. Okay. Let's see. Colony stimulating factors. This is one of those concepts that I could spend days and days on, but I'm not going to. It will come up again later. If you take immunology with Dr. Buck, I'm willing to bet he will spend some time on it. Colony stimulating factors relate to the differentiation of those white blood cells. Okay. Different kinds of white blood cells are stimulated by different colony stimulating factors. And that is what helps determine whether that progranulocyte becomes the basophil, the neutrophil, or the eosinophil. Okay, that's all you need to know about those. Okay, that's their job. That's all you need to know for my purposes. Now, in order to make red blood cells, we need a hormone. Told you we weren't done. The hormone that we need to make red blood cells is erythropoietin. EPO is how you'll see it very often referred to, right? Its job is to encourage the body to make more red blood cells. A typical, and I may be getting ahead of myself. Am I getting ahead of myself? Not too far ahead of myself. A typical red blood cell lives about 120 days. Okay, from development to the time it's broken down and it's recycled, about 120 days. Okay, if your body has some sort of blood loss or low blood pressure or something where there's an issue, this erythropoietin hormone will be released and your body will start increasing the production of red blood cells. Okay. Do you have any questions about this? Do we feel a little better about this? This is just a table that gives you all of this. Don't stress this column. You should have a general idea of the functions of what these guys do. We're going to talk in a little more detail about some of them than others, but If you're going to be asked to identify which ones are which, that will happen in lab, not in here. Okay, I'm not going to give you a picture of a white blood cell and say which white blood cell is this. Okay, that's a lab thing where you can do it with the actual slides. Any questions about that? Okay. All right. So red blood cells. First of all, red blood cells are unique in the body in that they lack a nucleus. Okay, they are anucleate, and they have a biconcave disc shape, which means they are almost a donut, right? If you just squeeze just a little bit harder in the middle, then you have a donut, right? Make an actual hole. They are very flexible and bendy, and they have to be flexible and bendy because your vessels that they travel through vary in size, from little teeny tiny capillaries all the way up to great big arteries, right? And so when they are going through capillaries, they have to bend and sort of twist to get through there. They contain hemoglobin, and hemoglobin contains a pigment that is the reason why they are red. Okay, hemoglobin makes the blood red. They also contain some lipids, some ATP, and something called carbonic anhydrase, which is going to be very important later. But we're going to talk about it here in a minute. But it's going to be really important again in a couple of weeks, actually, a couple of days, and in both the digestive system and in the respiratory system. We will talk about carbonic anhydrase. Now, here's the thing. If I say to you, without reading my PowerPoint, what is the job of red blood cells? What's the first thing that pops into your head? Transporting oxygen. How many of you ever even consider the idea that it also transports carbon dioxide? Okay, this is one of those things that most people are like, oh, transports oxygen. It doesn't have anything to do with carbon dioxide. That is not true. It does both. Okay, now, transporting oxygen and carbon dioxide is a really important task of the cardiovascular system. It is done in different ways for each gas. Okay, the majority of your oxygen is transported via the red blood cells through hemoglobin. They attach to the hemoglobin molecules, and the red blood cells carry them to where they need to go. Okay, most of the oxygen gets carried that way, not all. There is a small percentage of oxygen, 1.5%-ish, that dissolves in the blood and is transported that way. It just kind of hangs out in the blood. Okay, this is very different from carbon dioxide. Okay, carbon dioxide can also be dissolved in the plasma, slightly larger percent, 7%. You don't need to know the numbers, but again, you should know the relationships, right? I'm less concerned about specific numbers that you can always look up, and more concerned about you actually understanding the relationships to these things. Okay, so 7%, the smallest percentage of carbon dioxide, is dissolved in the plasma as carbon dioxide. Okay, a slightly larger percentage, 23-ish%, is actually transported by the red blood cell attached to hemoglobin. The largest percentage of carbon dioxide in your body is transported as bicarbonate in the bloodstream. Okay, which means that we have to take that carbon dioxide and we have to convert it into bicarbonate. And we do that through a reaction that uses an enzyme, carbonic anhydrase. And this allows us to combine carbon dioxide with water. Okay, and that is going to end up producing carbonic acid. And then the carbonic acid is going to lose its hydrogen, and the rest of what's going to be left is that bicarbonate. Okay, I have described the reaction to you. You do not need to know the details right now. Those details will happen in the respiratory system. Okay, for now, you need to understand that carbonic anhydrase is responsible for the conversion of carbon dioxide to bicarbonate. and that bicarbonate is the primary way that we transport carbon dioxide. Believe it or not, in terms of regulating your breathing and your respiration rates, carbon dioxide is significantly more important than oxygen. Okay? Your breathing rates are much more sensitive to levels of carbon dioxide in the body than they are to oxygen. Okay? The body works really hard to control carbon dioxide because carbon dioxide affects pH. And a tiny, tiny shift in pH one way or the other will kill you pretty rapidly. Alkalosis, acidosis, either way you die. Okay? It's always entertained me when people try to drink alkaline water and increase their alkalinity. Your body's not gonna let you do that. Questions? No? No? Am I spending time on things we don't need to spend time on?
So the red blood cell is always carrying carbon dioxide in that form, is always carrying it as bicarbonate ion.
No, the red blood cell is always carrying carbon dioxide. The bicarbonate gets dissolved in the plasma as bicarbonate. So the plasma is going to carry bicarbonate ions and carbon dioxide ions.
Okay, but the red blood cell.
The red blood cell is carrying carbon dioxide. Yes. The production of bicarbonate happens in the red blood cell. So the carbon dioxide will come in, it will get converted within the red blood cell. Because remember, the red blood cell contains that carbonic acid, which is on a slide somewhere. Where it was. Sorry, the carbonic anhydrase is inside. Oh, there it is inside of the red blood cell, right? So that conversion of carbon dioxide to bicarbonate happens in the red blood cell, but then that bicarbonate is sent out of the red blood cell into the plasma. Yeah. Question. Anything else? Okay. All right. So we've talked about the hormone erythropoietin and that its job is to stimulate red blood cells. It does so through a process. This is another one of those beautiful things about how they name some stuff in the anatomy and physiology. The process of forming red blood cells is erythropoiesis. So the hormone erythropoietin drives the process of erythropoiesis, which makes new red blood cells. Okay. Generally speaking, this happens as a result of low blood oxygen levels. If your blood oxygen levels are too low, the first thing the body does is it increases the production of red blood cells because then you can carry more oxygen. Okay. I said 110 to 120 days. When the red blood cells reach their age limit, when they age out, they undergo hemolysis, right? They rupture. The hemoglobin that exists in the plasma after that rupture is then broken down by a macrophage. What is a macrophage? Big cells that eat things, right? Big cells that eat things. That's a macrophage. Macro, big, phage, to eat. Big thing, big cells that eat things. Okay. So when we're talking about producing red blood cells, the first thing that's going to happen is this detection of a drop in oxygen. Where are we going to detect that? In the kidneys, yes. So in the kidneys are then going to increase the production of erythropoietin. That erythropoietin is going to get sent to the bone marrow, and the red bone marrow is going to produce additional red blood cells, which hopefully will lead to the increased production of, or the increased availability of oxygen. Easy enough? Okay. All these systems are interlinked. Are you guys starting to figure that out? Everybody's interconnected with everybody else. All right, now our macrophage has discovered, well, we're not going to do that yet. We'll do that on the next slide. Okay, so hemoglobin. Hemoglobin is a protein. Okay, there are 4 subunits in this protein called globins. Okay, each little subunit is a globin. Now, when you guys took A&P 1, did you talk about protein structure? Primary, secondary, tertiary, quaternary? Okay. Do you remember the difference between, do you remember the 2 different kinds of secondary structures that exist? Let's start there. A beta pleated sheet and an alpha helix, okay? So I point that out just because 2 of these globin units are betas, beta pleated sheets, and 2 of these globin units are alpha helices. So what you have here is 4 tertiary structures, 2 of them that started as a beta pleated sheet, 2 of them that started as an alpha helix, that come together to form that quaternary structure, that functional protein of hemoglobin. Okay, I always like to point that out because otherwise students are like, why did we talk about that? This is why. Talk about that so I can talk about this. Okay, now in humans, there are 3 different kinds of hemoglobin. Why? First of all, what are they? We should be able to tell that easy. And why are there 3 different kinds?
Okay, so basically it's gonna be like 3, I guess, like, growth stages.
Okay.
So basically, embryonic,
Mhm.
and then fetal is like, after the born, right? But
Fetal is actually once your heart starts functioning. So you go from being an embryo to being a fetus to being a baby.
Yeah.
So, okay, so we've got the 3 types. Why are there, what's the difference? Just embryonic? Yes. So they don't necessarily require a great deal more oxygen, but there is something about oxygen. So what is unique about embryonic and fetal hemoglobin? A greater attraction to oxygen. 99% of the time, if I'm asking the question, the answer is probably on my PowerPoint slide, just for those of you who are panicking a little bit that I'm asking questions. So embryonic and fetal hemoglobin have a significantly higher attraction to oxygen. Okay, why would that be necessary for an embryo and a fetus?
developing everything, and it's kind of piecing it together in a sense.
Yes, but so technically so is a baby after it's born, but this particular kind of hemoglobin stops being produced after birth.
Is it because like 10 people is so
Say that again slightly slower.
okay, because it's so
Uh-huh. So you're on the right track. It's not about sharing hemoglobin, but sharing something else, oxygen. Where does a fetus and an embryo get their oxygen? Not the mother's hemoglobin. It comes from the diffusion of oxygen across the placenta. Okay, so how much oxygen is happening there? A lot? No, right? How much oxygen is dissolved in the blood? How much oxygen is dissolved in the blood, in the plasma? 1.5%, right? If it's not bound to the hemoglobin, right? Talking about oxygen itself diffusing across the plasma membrane. Small percentage of available oxygen, right? If all of the oxygen was going to the fetus, the mother would die, right? Mom's got to stay alive. She's the incubator. She's got to stay alive or the baby doesn't make it. So in order for the fetus to have as much of that available oxygen as possible, the fetal and the embryonic hemoglobin are more attracted to that oxygen. So even at very low levels, that hemoglobin is picking up all of that oxygen. This is something that happens with species that are adapted to live in high altitudes. Their hemoglobin becomes more sensitive to the presence of oxygen because oxygen is at such a lower level at high altitudes. And so this is what happens, right? Once the fetus is born, production stops after birth. Why? What's the change? They're able to breathe, right? When they come out of the vaginal canal and they get whacked on the bottom. I don't think they actually do that anymore. They used to hang you upside down by your feet, whack you on the bottom. I'm pretty sure that's what happened to me. So when they do that and they take that first big scream, what do they have to do before they scream? Right? That inhale does a couple of things. It closes a hole in the heart that we'll talk about when we get to the cardiovascular system, and it expands the lungs. And it allows the pulmonary circulation to start working. And therefore, they no longer need the production of that higher hemoglobin. And because now they've got actual oxygen coming in that they can use the regular old normal hemoglobin for, and that's all they need. Make sense? Kind of cool. Okay. Now, hemoglobin is a funny little molecule. It can carry oxygen. It can carry carbon dioxide. It can carry neither. If it's empty, it's just empty. Okay. Now, because of that, a single hemoglobin molecule can be called a number of different things. Okay. So if the hemoglobin molecule has oxygen bound to it, it would be oxyhemoglobin. If the hemoglobin has given up its oxygen and it's just hanging out in the world, empty and naked, what are we going to call it? Deoxyhemoglobin. Once it picks up the carbon dioxide, it becomes carbaminohemoglobin. Now here's the dangerous one. If the hemoglobin picks up carbon monoxide, what does it become? Carboxyhemoglobin. Now here's the issue. Carbaminohemoglobin is sort of like if you imagine getting into a taxi and being driven to the lungs, and then the taxi opens a door and the carbon dioxide gets off and goes out into the lungs, and the oxygen gets in the taxi and goes back out to the body, right? That's basically how this transition works between oxyhemoglobin to deoxyhemoglobin to carbaminohemoglobin, right? And that's a beautiful process, right? We get in, we get out, we get in, we get out. Carboxyhemoglobin is more like on Criminal Minds when the guy gets in the back of the taxi cab and the doors are locked and he can't get out anymore. Nobody else can get in, he can't get out, and you got problems. Okay? Carboxyhemoglobin reduces the availability of oxygen carrying capacity. That was a lot of words that I think got my point across. It also reduces the body's ability to get rid of carbon dioxide. If you get a lot of carboxyhemoglobin buildup, you die. Called carbon monoxide poisoning. Now, I am not a medical professional. Are any of you like paramedics? I usually have at least one paramedic in the room, and they usually correct me when I do this wrong. So I had a paramedic last fall who told me this, and I did not know this, which is why I don't have medical experience or people's lives in my hands. Apparently, there is a point at which there is too much carboxyhemoglobin in the bloodstream. And if you give that person oxygen, you actually increase the rate at which they suffocate.
Yeah.
Well, it prevents you can't, yeah. And so you die faster. You give them oxygen thinking you're going to help them and you kill them. This is why I don't want anybody's life in my hands. I don't need that. I don't need that in my, because I would take that home with me. Okay, questions? Y'all gonna go home and check carbon monoxide detectors? Stuck. Once it gets there, it's there. Yep. Can't get it back out. Yeah, it stays there until the cell undergoes hemolysis, and we're going to talk about that process now. Because at some point, the cell's going to die, right? 120 days. Could you imagine if you knew you only had 120 days to live, what you would do with your life? I mean, I would have a good time. I would have a really good time. Right? So when the red blood cell dies, that macrophage is going to come in. All right, so your red blood cell undergoes hemolysis and it goes poof, right? And now we've got all this cell debris floating around, right? Including the hemoglobin molecules. Macrophage comes in and picks up those hemoglobin molecules and breaks them down. Okay. Now, what's going to happen is a couple of things. The globins, those alpha beta protein structures, those are going to get unwound. They're going to get denatured. They're going to get unwound, and they're going to get broken down into their subunits. What are the subunits of proteins? Amino acids. Yes, they're going to get broken down into their amino acids. Those amino acids can then be taken by the body and reused to make more protein, right? The body is really good at not wasting things. The iron that is part of the hemoglobin, that is necessary for the production of hemoglobin, primarily is going to be taken to the red bone marrow, and it will be recycled and used to make more hemoglobin. Okay, so that leaves us with the trash, right? The pieces of cellular debris that we can't recycle iron because they're not iron, and we can't recycle because they're not amino acids. We've just got garbage we got to get rid of, right? We're going to do that by converting those leftover chemicals and pieces to something called biliverdin. Okay. Now, biliverdin, through a chemical process, is going to get converted into something called free bilirubin. Okay. Free bilirubin is going to get combined with something called glucuronic acid, and it is going to become conjugated bilirubin. Okay. The reason we need to do this is because conjugated bilirubin is more water soluble than free bilirubin. And our goal is to combine this conjugated bilirubin in the digestive tract with bile so that it can then be removed from the body. Okay, so that conjugated bilirubin gets added to the bile, and most of it gets excreted out of the body in the feces. Okay, there is a small amount of bilirubin that always escapes the fecal process and ends up back in the bloodstream. That gets filtered in the kidneys and is removed from the body through the urinary process. Okay, so here's the thing. There are two different structures that we have, or chemicals, I guess. We have urobilins and we have stercobilins. Guess where I'm going to find your urobilins? In the urine. And stercobilins in the feces. Those two guys, urobilins and stercobilins, are the reason for the color of your feces and your urine. Fun facts. Share them at the Thanksgiving dinner table. I'm sure the family will be totally impressed. They loved it the year I came home after taking ornithology and proceeded to identify all the bones in the turkey. They were like, really? All right, any questions about this? Feeling pretty good about this? Yeah, okay. All right. We're gonna go pretty quickly through the white blood cells, right? Because we're gonna talk more about them again a little bit when we get to the lymphatic system. You guys will spend or have spent, have you guys done this lab yet? The blood lab? Did you look at blood cells in the blood lab? Yeah, that's right. So you guys already done this in the lab. I'm not gonna spend a ton of time here. I literally have, I think there's 2 slides. Yeah, 2 slides. And literally what you need to know are the bullets that are on these slides. Right? That's all I'm going to ask you about this stuff. Are there any questions about any of the terms on this slide? I don't want to spend time here if we don't need to, but because we're going to need to spend time in the next section. So we understand chemotaxis, diapedesis, amoeboid movement, and phagocytosis. Okay, moving on. Again, you just need to know the bullets here, right? This is really basic, straightforward. What do each of these cells do? Any questions I ask about these kinds of things will be related to material on these slides. Easy enough? Anybody have any questions about any of these? No? Okay, moving on. This is where we generally run into trouble. I'm gonna make this go away. Hang on. Go away. Okay, so platelets are responsible for what job? Blood clotting, okay? Generally speaking, blood clotting, the process of blood clotting is a process called hemostasis, right? Trying to manage the blood, maintain the blood, keep it at the same level. There are technically 4, although I don't spend a lot of time talking about clot retraction because it's sort of everything's done and we're going to try to get rid of the issue. There are 3 real phases that we're going to spend some time on here. But it's important that you understand is that they are not consecutive phases. Okay, they are concurrent phases. So if I were sentencing you to jail, you would be serving all 3 phases at the same time rather than one after the other. Okay, we much prefer concurrent. I spent some time on the grand jury recently. So that would be why all of this criminal discussion keeps coming up in my, plus I love crime TV. Okay, so we have our vascular spasm, which is the start of the process. Vascular spasm will last the entire length of the process. Okay, Then we move into the formation of the platelet plug, and then we move into the formation of the clot or coagulation. Okay. Now, I have provided you with here at the bottom of this screen pictures from an old textbook. And I've given you these because there are some things in the new textbook that I like better. And there are some things in the old textbook that I like better in terms of the visual explanation. And so I've given you both. So you have them if you want to refer back and forth. When we talk about the vascular spasm, so first of all, it's important that you remember what we're talking about here. We're talking about some issue that has cut into or opened up a blood vessel. Okay. The first thing that the body is going to do when a blood vessel is opened is it's going to contract the smooth muscle in the walls of that vessel. Because what? What is the because at the end of that statement? It's going to slow the blood loss, right? The smaller the opening, the less blood can get out at a time. If we just leave it open, it's just flowing free, right? Now, this only works to an extent, right? If I have a paper cut, right, I'm talking about a much smaller vessel than, here we go again, knife to the aorta, right? It's vastly different structures, okay? Vastly different sizes of wounds. Vascular contraction is not going to help you from bleeding out if somebody's cut your aorta, okay? It's just not. So when that damage happens to the vessel, we're going to have that spasm, and there are going to be chemicals that will be released from the damaged cells. And those chemicals include endothelin and thromboxanes. The thromboxanes are actually going to be released from the platelets involved in this process. The endothelins are going to be released from the actual cells that were damaged. Okay, that's an important distinction. Now, the vascular phase lasts about 30 minutes. So that vessel will stay contracted, reducing blood flow for about 30 minutes. This allows the body time to repair the damage, or at least to start repairing the damage, okay? There are going to be a couple of things that are going to happen, okay? So first of all, when the endothelial cells contract, there is exposure of the basement membrane of those endothelial cells to the blood. This is not a normal thing. Normally the basement membrane is covered by the endothelial cells, but when they contract, when they get pulled, there's exposure, okay? The endothelins will cause that continued contraction, and they will encourage the beginnings of cell division. Sorry, my brain lost that word for a minute. They will encourage the process of cell division to start, because we got to do that, right? The only way to fix a hole is to make more things to put in it. We're also going to produce some ADP. We're going to release some tissue factors, which we will talk about later, although you don't really know all of them, and something called prostacyclin. Okay. The other thing that's going to happen is that the plasma membranes on the endothelial cells, the cells that are on the inside of the capillary or the blood vessel, they're going to get sticky. Okay, they're going to get real sticky, and that's important. Now, are there any questions about the vascular phase? Because that's really, it's pretty straightforward. Yeah. I'm going to explain to you in just a minute. That's yeah. So the sticky is going to relate to the next process. Okay. All right. So this is the platelet phase. Okay, the formation of the platelet plug. Now, I want you to look at this image. This is the new textbook, I think, and I really like this image. So I've got first of all, orient yourself, right? Because everybody looks at this and thinks outside, inside, but it's not, right? This is your smooth muscle in the wall of the vessel. This is your smooth muscle here. This is the vessel wall here. This in here is the lumen of the blood vessel. That's where the blood is. Okay. So we've got a tear in that vessel that has happened, and it is exposed the collagen. Let's try a color we can see here. The collagen and the smooth muscle. Okay. These are your endothelin cells in here, the endothelial cells. Okay. So those endothelial cells become sticky and they start to sort of attach to things. And on top of that, we have these little guys. Where's the word? Find the word so I can point it out to you. Oh, it's not in that list. It's over here. These little guys here, the von Willebrand factors. Okay. These von Willebrand factors, you see them here. They're these little black, they look almost like little stitches. Okay. Think of them like Velcro. Okay. When I say sticky, that's what I mean, right? It makes it so that I can attach things. Okay. So the von Willebrand factors and the stickiness associated with those endothelial cells, that's what I'm looking for, allow those platelets that are coming in to attach. Okay. They attach to the walls. So you see down here, this one's attached to the smooth muscle. These are attached to the cartilage. This little guy is going to be attached up here to this endothelial cell. And they form that initial layer. And when they do, they release thromboxane. Now, thromboxane causes chemotaxis. What is chemotaxis? It's attraction, right? It's basically they send out the alarm to all of the other platelets in the vicinity and say, yo, guys, we got a leak. We got to plug the leak. And so it attracts and brings in additional platelets. Those additional platelets attach themselves to the existing platelets, the ones that are already on the torn area. Okay. And they will continue to do that. Every time a platelet attaches to this forming group, they're going to release more thromboxanes. Those thromboxanes are going to call in more platelets, which are then going to bind, which are going to release more thromboxanes. What am I describing here? Positive feedback. Okay. Blood clotting is a positive feedback process. Now, this will continue basically until the vascular spasm stops and the blood has stopped flowing. So the thing I want you to notice here, because this is going to be important in a minute, is that on each platelet, I have these little structures that are fibrinogen receptors. Okay. And the thing that holds those two together is fibrinogen. Okay. Fibrinogen is going to become really important to us here in a few minutes. Okay. So we have our platelets forming our platelet plug. This is not a blood clot. This is just a platelet plug. It is the structure, it's the framework that we're going to build the blood clot in. Does that make sense? Yeah? Okay. So, moving along. Any questions before I move along? Yeah. Chemo, CHEMO. Yeah, it was back here somewhere. Here. Okay, now, this is where I am being very nice to you guys because I'm giving you another version of that. That's the one in your book. This one we're going to use. This one is much better. Okay, so this is phase 3. This is the coagulation phase. You guys don't have this on this slide in your PowerPoint. It's on that slide back at the beginning of this process in case you're looking for it. I moved it here because we're going to use it and it's better to move it now than later. So here's the deal, okay? We have a fairly complex process that is going to happen here. And it's going to happen starting with it's going to start as soon as the vessel gets cut. Okay, remember I told you these are concurrent processes. So the vascular spasm is happening, the platelet plug is developing, and coagulation is happening. Okay, so coagulation involves 3 pathways that all work together to generate a blood clot. Okay, and all of the words are here, and you can look at the words. We can talk about them later, but I want to look at this image, okay? Because this image really does a nice job of laying this process out. So what I have here, just so we're all on the same page, right? I have my smooth muscle. Come here. I have my smooth muscle, I have my cartilage, I have my endothelial cells, and this is the lumen of the red blood cell, of the blood vessel. Okay? So all of this stuff is happening inside of the blood vessel. Okay? When this cut happened, okay, 2 things resulted from this cut. First, there was tissue damage. Okay? There was physical tissue damage. As a result of that tissue damage, we had the exposure of the collagen. Okay, so this collagen that got exposed when that tissue damage was done, that's the second trigger. Okay, so 2 separate triggers that are working here. Okay, they're working simultaneously, right? This is one of those processes where it's like I'm giving it to you in steps, but it's all happening at the same time. So it makes your brain hurt a little bit. Okay, so cut the vessel. I have tissue damage. I have exposure of cartilage, right? Vascular spasm is happening. Platelets are starting to come in and build the platelet plug. But in the process, because this tissue was damaged, the extrinsic pathway was triggered. And because this cartilage was exposed, the intrinsic pathway was triggered at the same time. Okay? These work all at the same time. Now, I am not going to ask you to identify all of the individual factors and what order they come in. Okay? The things that I really care about you knowing are, I'm going to identify for you here. So you need to know that tissue damage leads to the activation of the extrinsic pathway. Okay? Don't worry about factor 3. Don't worry about calcium. Don't worry about factor 7. Okay? The damage of the tissue leads to the creation of a tissue factor complex. And that tissue factor complex is going to lead to the production of factor 10, factor X. Okay? They're Roman numerals, but I like factor X better. It makes me feel better. So the extrinsic pathway produces factor X, factor 10. You guys with me so far? Have I lost anybody? Okay. Now, at the same time, the exposure of the collagen has triggered the intrinsic pathway. And that is going to cause a bunch of proenzymes to be activated. Right? We've got these proenzymes, these enzymes that are going to basically become an enzyme. They get activated. Okay. And we have platelet factor 3. This is the only one you need to know other than factor X. And the combination of platelet factor 3 plus the activated proenzymes, we can ignore calcium, we can ignore those two, will create an activator complex, a factor X activator complex. What do you think it does? Activates factor X, right? The extrinsic created it, the intrinsic turns it on. Yes. Platelet factor 3. Yeah. Yes, the combination of platelet factor 3 plus the activated proenzymes will create or form the factor X activator complex. Yeah. The exposure of the cartilage of collagen. Sorry, the exposure of the collagen. Yeah, it's a very tiny difference, right? You've got the damage and the exposure. The physical damage is extrinsic. The exposure is intrinsic. Okay. Now, I have now managed to get to an activated factor X. This is where things get fun. Okay. Factor X is a prothrombin activator. Okay. It's its job. Okay. What that means is it takes prothrombin and converts it or activates it, turns it on, and makes it into thrombin. Why do I need thrombin? It converts fibrinogen into fibrin. Where is the fibrinogen? In the platelet plug, right? It's that connector that's holding those platelet plugs together. When thrombin converts fibrinogen into fibrin, that is when you get your clot. Okay. It's released in the process somewhere, but we're not going to go into that level of detail. Yeah, it's released in the process somewhere. But yeah, and we're not going to go that level. There's a lot of little things that get released in this process, but it's not necessary to understand the overall process. So this is how we get to the actual clot, right? This is not how this works, right? This is the cartoon in my head. Okay. In my head, fibrinogen being converted into fibrin is sort of like cheese melting over macaroni, right? Where they'll start to stick together and it becomes a solid mass. Yes, fibrin you can think of, yeah, kind of wrapping around it and holding it all together. Yeah. And so whatever works for you is great. The end goal is that we end up with this clot that seals off both ends of the vessel so that the blood flow stops. Vascular spasm will then relax, and then cell division will start to take place and they'll start to repair that vessel. And in the process of that happening, we will have clot retraction. And clot retraction basically happens when we start to break down that fibrin. It gets smaller and smaller, it pulls away. Think about a scab, right? It pulls away until it gets smaller and smaller and smaller, and then eventually it just goes away. Okay. And that is because we're healing, we're repairing that tear underneath a little at a time. Does that make sense? So scarring is related to, was this last semester? Maybe one. Well, it's that, or, so you guys learned about dermatomes? You didn't learn about dermatomes? Some of you did, some of you didn't. Some of you did, some of you didn't. So dermatomes are basically, it's in your textbook. If you want to go back to the integumentary system, you can go back and look at it. You can basically map the human body in terms of the orientation of fascia, and yes. And so cutting with the dermatome and stitching with the dermatome will reduce scarring. Cutting against, because you're getting pulled in the wrong direction, will increase scarring. So plastic surgeons are very good at figuring out how to make the cut look nice. Yeah, that's basically. And there are other issues. Keloids happen, and you know, scars can become. Some of it is post care, right? Taking care of the scar, or taking care of the incision properly afterwards. Massaging it properly will help reduce scarring. There's a lot of things. Sorry, what? Well, yeah, so it's got a lot to do with like she said, how movable the area is, right? Because the more movable the area is, the more stretching force you're putting on that incision, and the harder it is for these poor little cells to divide themselves. They're like, would you just hold still? It's exactly what happens. Yep. Start over again. I gotta start over again. Yeah. Elbows, wrists, ankles, knees. I've got a total knee replacement scar that runs from the top of my kneecap to mid shin. That was the 4th surgery on that knee. I had a PCL replacement, an ACL replacement, a medial meniscus tear. And then in 2014, I finally reached the point that I needed a total knee replacement. And when he went in, because he didn't want to do it because I was too young, because they only last about 20 years, which means I'm probably going to have a revision at some point. But when he went in, he said that had they waited 6 months, they would have had to have done bone grafts before they'd have been able to do the replacement, because I had a bone spur that had grown up in between the condyles of the femur. And so it was just constant grinding on that. And so he had to, because I had a bolt in my knee from when they replaced my PCL. And he actually had planned on just leaving it there, but in order to get good grip with the tibial insert, he had to open the incision farther and go further down into the bones. They had to take it. They ran it through behind. It was a very complicated thing. It was awful. And actually, what's funny is that when he replaced my knee, the PCL and the ACL were both gone. Both had been wiped out by the bone spur. And now I don't have one because you can't put one in with an artificial knee. Yeah. It's funny though, because my knee sets off the security thingies.
What you got?
When I was on the grand jury, I had to go through security every day. And I'd walk through and I'm like, it's the knee. And they'd take the wand and like, it's the knee. It's right down there in her knee. All right. Yeah, right. I was doing competitive powerlifting at the time when I had the knee replaced. I wanted him to put hydraulics in.
You're basically cheating.
Huh? I started in 2010 and I did my last meet in the spring of 19. 9 years. Yeah. And I might get back into it at some point, but COVID hit and the gym was a nightmare, and it was just one of those things. And then I got out of the habit, and but it was fun. It was a lot of fun. My mother was a competitive powerlifter too. She started at 64.
Oh my God. Wait, what?
It's never too late. It really isn't. Yeah, she started at 64. Actually, she just turned 75 and she wants to do another meet because she's got the 60 to 64 titles and the 65 to 70 titles, but she missed the 70 to 74. She wants the 75. So yeah, so all of the Kentucky State titles in those age ranges, my mother has them. She actually has a couple of world as well in the USBA. I have one world deadlift record, and a bunch of state and local stuff. That's fun. It's a lot of fun. If this were MP1, I would show you a video of my mother at some point, but it's all right. It's all right. I have squirrel brain. We're almost done, so we'll be out of here early. Okay, clot formation, anticoagulants. All you need to know is what they do. Okay, and I don't know why I leave this one in here, because don't ever ask anybody about it, or that one, sodium citrate. So anticoagulants prevent coagulation factors from starting clot formation. Okay, so basically we're gonna affect those factors, the ones I told you to need to know the names of, right? Factor 1, factor 2, factor whatever. Coagulation occurs when those factors exceed a particular threshold. Okay, once that threshold is exceeded, this is not a threshold like action potentials. It's not an electrical threshold. This is a physical quantity threshold. Okay, when we get so much of these factors in the bloodstream, that is when your coagulation is gonna start happening. Couple of things that we have antithrombin, which is going to turn off thrombin. We have prostacyclin, which I mentioned earlier, and it comes from the endothelial cells, and it causes vasodilation, which is going to do what? It's going to reverse the spasm, right? It's going to open that blood vessel up, and it's going to inhibit the release of clotting factors from the platelets. Okay. And then there are things that you can take that will also reduce clotting. Heparin being one of the most common, warfarin being another. There are a lot of different ones that are available out in the world now. I only care that you recognize that heparin is an anticoagulant. Okay. Clot retraction, we talked about this briefly. Fibrinolysis undoes fibrin, right? It breaks down fibrin, which is going to help dissolve that clot. Okay. Plasmin actually takes that fibrin and hydrolyzes it. What does that mean? Adds water, right? Hydrolyze is a derivative of hydrolysis, which is a reaction that breaks down things by adding water, right? So anytime you see hydraulic or hydrolyzes or hydrolytic, you'll see a lot of variations of that. That's all hydrolysis. Make sense? Okay, you guys did blood typing in lab. I put this here because sometimes there are additional questions or there's confusion about this stuff. Are there any questions about blood typing, or do you all kind of get it? That was not a yes or no question. Presence of what? I'm going to use the words.
I was very confused with that last because it was saying that like whatever it needed was like antibodies.
Okay.
But where did
I
you
know-
just suck on the-
Okay, hang on. Let me.
Like are the things that attack the whatever. Like, I was thinking of in terms of blood transfusion.
It's the other word I'm looking for. Antigen, antibody. Oh. All right. Antigen, name tag. Okay. Because they're going to come up again, right? Antigens are a big thing in the lymphatic system, in the immune system. Antigens don't mean good or bad. Antigen just identifies, right? So if I have a blood cell, which is of course green because we're all aliens, and on that blood cell, I have antigens. Now he needs a smiley face. Sorry. I couldn't nod. He had ears in the throat. He had to have a face. Antigen A. Okay. That means that I have type A blood. Okay. His name tag. His name tag tells you what he is. Okay. Now, in the bloodstream, because I have type A blood, I cannot have what? Well, in my body, be antibodies. No, hang on, back up. I have type A blood. I cannot have type A antibodies. Okay. If I have type A antibodies, my antibodies are going to attack, right? So the antibody is always looking to attack the thing that is similar to its name. Okay. So in this particular individual, I would have try using the, there we go, anti-B antibodies. Okay. Which means if I transfuse with somebody who has B blood, so if I put a blood cell in here that has B's, it's a compulsion. I can't help it. If I put somebody in who has B's, right, this antibody is going to attack. And it's going to take all of those B blood cells and it's going to clump them together via agglutination and cause a clot. And if you've transfused a lot of blood in that way, you will kill somebody. Okay. Now, there is a bit of a caveat to this process when it comes to RH antibodies. Okay. Or well, RH antigens. Okay. So if I have an individual, we'll do this in a way that doesn't require me to draw faces. Let's make it purple. Okay, if I have, no, now I need, sorry. It's gotta happen. So there's just no way to do it. You will find that RH is often identified as D. So you'll see them used interchangeably. Okay. If I have this individual, so they are this is what I got, that would classify this individual as A positive, right? The positive and negative references the RH factor. Okay. If I am A positive, then I cannot have what? I know I can't have, well, let's do this another way. What antibodies exist in this blood? Anti-B. Anti-B. My colors are getting all out of whack here. Anti-B. Okay, I can have those. I can't have the anti-RH. If I have the anti-RH, it will attack my blood. I can't have the anti-A because it will attack my blood, right? Now, I'm gonna shrink. That's why I love this app. You can just indefinitely make more room. So if I have, individual, ooh, that's a bad circle. That's better. And on this individual, I have this. And that's all he gets. Actually, let's do, no, let's do it this way. Okay, that's all he gets. This one would be A negative. This is an A negative individual, right? What kind of antibodies do I have here? I have anti-B. Maybe. Maybe. So here's the situation with RH. If you are negative, you do not automatically have anti-RH antibodies. The only time you develop them is if there is exposure to positive blood. Okay. So if this person managed to expose this person to their blood, then they would develop the anti-RH. Yes, yes. There has to be, this is basically an immune response. This is actually a really nice lead in to what we're going to talk about on Monday. Because this is an immune response, right? It's like any other foreign antigen. The body's like, you don't belong here. And therefore, we're going to create a defense against you. Okay. So here's how this works. Here's where this becomes a problem. Hemolytic disease of the newborn. Okay. This is a problem for a RH negative mom who is carrying an RH positive child. Okay. But the effect of this is actually only going to impact baby number 2 or 3, etc. Okay.
If the baby is RH negative, right?
If the baby is RH negative,
Like the second or third child, right?
yeah,
Because the mom.
no, the baby, if the child is RH positive, and here's why. Okay, because remember that the RH positive child has a blood cell that has, we'll just say A, D, A, D, A, D. They have the antigen, right? So if during delivery, or sometimes it just happens randomly across the placenta, some small amount of blood from baby 1 gets into mom's body. Mom's body is then, after the delivery, is then going to develop antibodies. Okay. After baby 1 is delivered, mom develops antibodies. If baby number 2 is RH positive, and any of mom's blood that now has antibodies in it gets transferred to the baby, then that blood will kill that baby. Okay, there is a way to prevent this. It's called RhoGAM. RhoGAM. And this is why blood tests are so important before you start having babies. If you are an RH positive mom, an RH negative mom, and dad is RH positive, then you want them to give you RhoGAM during each pregnancy, because RhoGAM basically prevents the development of the antibody. So before every pregnancy, or at the beginning of every pregnancy, you get this shot, and it prevents the development of that antibody so that you don't kill your baby.
And like, okay, and I looked at RH negative is like this as a gene.
Okay. I don't know. You're I don't know.
But would this happen with like an RH positive one and an RH negative child?
No. Why?
Or is this even have-
Mhm. Exactly. Only goes one way. Yep. If you're RH positive, you want babies, you're fine. You're fine. We don't worry about you. Okay. All right. These last 2 slides are quick and easy. They are literally just memorization. Terminologies, definitions that you need to know. Okay. Numbers. No. Don't memorize the numbers. You do not. So this is generally my theory on this, right? If it's a number and you need to know the exact number, you're not going to need to know the exact number at a split second. You're going to be able to look it up, right? But if the doctor says, I need a red blood cell count, and you're like, well, I don't know what that is, you're going to look like an idiot. Okay. So we try to prevent that from happening. Right? We don't want you to, we don't want that to happen. I would like for you guys to be like, yeah, I'm from Pana, Corpus Christi. And they're going to be like, yes, you know your stuff. So know the terms, know the definitions. Don't worry about memorizing the specific values. Do you understand the difference between blood typing and cross matching? And why both need to happen? So this is a common misconception that ABO, RH are the only potential issues when you're donating blood to somebody, right? There are hundreds of antigens on your blood cells, okay? Any one of them could create a reaction. So when you are getting transfused, they do a blood type, and then they take your blood and whatever blood it is that they're planning on transfusing, and they mix them to make sure that there's not some kind of a side effect. There's not some kind of a reaction. So it's not just oh, this person's B negative, so we can give her any B negative blood that happens to be in the bank. It's here's this B blood, let's test it and make sure that it works. Okay. Usually those questions are about the potential that you've picked up some sort of a pathogen that is not native. Yeah, because we don't want to be bringing those things in. Okay, other tests. More lists. Differential white blood cell counts. Again, don't worry about the numbers. Make sure you understand what a platelet count is, and what thrombocytopenia is. Thrombocytopenia being a reduced number of platelets. Prothrombin time measurement, what it is, blood chemistry, all of those things. And then in terms of diseases, you should have, and don't worry about the descriptions, okay? Just focus on diseases that are listed under those categories, okay? Makes sense? Are we feeling good? Are you excited? Are you gonna be excited in like 2 minutes when I say go away? Go away. You guys are good. I'll see you guys on Monday. Oh, the multiple choice for your exam will open tomorrow morning at 12:01 a.m. You can take it anytime between then and 11:59 p.m. on Monday. Whatever makes you happy. If you wait till the last minute, it's liable to lock up on you because everybody else will wait till the last minute too. Just saying. Focus on the concepts where I spent a lot of time.